let's talk about the heart. We're going to start today with this adorable cartoon. Yes, again, it's from the Awkward Yeti. I know I always use the Awkward Yeti. I love the Awkward Yeti. It's the lungs talking to the heart, and the lungs say, "We got you something," and it's oxygen, and the heart says, "I love it, I'm going to share it with everyone," which is the job of the heart. Why do you have a heart? To move nutrients and oxygen through the body. You need to get oxygen to all of your tissues and then remove waste from all of those tissues. And then you need to get nutrients from the digestive system to all of your tissues. So today we're going to talk about the heart's role in that. And then next time we're going to talk about the circulatory system. We'll start getting into the role of the rest of the circulatory system in moving things around the heart. So the way the heart does its job is first, it gets oxygen into the blood. So the blood coming into the heart is low in oxygen, and it goes to the lungs where it gets oxygenated, and then it goes back to the heart and then goes to the tissues. Notice here that the blue is the deoxygenated blood, the red is the oxygenated blood. And oxygenated blood is actually more red than deoxygenated blood, and that's because the oxygen binds to iron in the hemoglobin molecule, which of course you remember from the last lecture. And when iron is oxygenated, it becomes reddish, like, rust. And then when it gives up that oxygen, it goes back to a darker color. And so that is why the deoxygenated blood is much darker. It's not actually blue, but it is darker because it doesn't have the oxygen turning the iron bright red. All right, so let's start with the location of the heart in your chest. Please notice that your heart is not located on the left side of your chest. It's in the middle. It's right behind the sternum. That's what the sternum's for. The sternum protects the heart. Nice big bone in the middle of your chest, protecting the really important pump in the middle of your chest. It does sit slightly left, especially the apex of the heart, this pointy end right here, which does project to the left. And we're actually going to have the left lung is going to be slightly smaller in order to make room for this apex of the heart. The right side of the heart sits on the front, the left side of the heart sits behind. The base of the heart is this area here, the superior border, formed by the left atrium. Think of the heart as a triangle upside down. The base is here and the apex is here. The superior border is where the aorta leaves the heart, where the pulmonary trunk leaves the heart. We'll come back to all of these. Don't worry if you don't know what those are. The conical bottom is the apex, and then the inferior border is the edge of the right ventricle. Okay, this is what it looks like in a cadaver. And you can see that most of this tissue here is the right ventricle. The heart sits so that the right ventricle is in the front and the left ventricle is primarily behind. This is the pulmonary trunk, carrying blood away from the right ventricle to the lungs. This is the aorta, carrying blood from the left ventricle behind, and that's then going to branch and carry blood to the whole body. So what differences do you notice, just looking here, between the atria here, atrium is singular, atria is plural, and the ventricles here? Well, it's pretty obvious that the walls of the atria are very thin and the walls of the ventricles are much thicker. And that's important because the atria don't have as much muscle mass and can't make contractions as strong as the ventricles can. When we look at the right ventricle here versus the left ventricle, especially if you look at this outside wall of both ventricles, you notice that the right ventricle has a much thinner wall than the left ventricle. And again, this makes sense because the right ventricle is pushing deoxygenated blood just to the heart, to the lungs. Excuse me, they're just going to here and here. The left ventricle, on the other hand, has to push blood to the whole body, including all the way up to the head, way up there. So there's physical differences because you remember that form and function go together. All right, so let's talk about the blood flow into the heart. The blood is going to flow into the heart from the superior vena cava and the inferior vena cava. This is the superior vena cava, this is the inferior vena cava. And just remember, all the tubes that are blue on any diagram or model, they are going to be carrying deoxygenated blood. Everything red is going to be carrying oxygenated blood. The superior vena cava is going to carry blood that has come from the head, the neck, the arms, and the very top of the trunk, basically the shoulder area. The inferior vena cava is going to carry blood from the rest of the heart. Everything below basically the shoulders is going to be, all of that blood is going to be carried through the inferior vena cava. This blood is high in carbon dioxide and low in oxygen. So the inferior vena cava comes in here, the superior vena cava comes in here. They both are going to dump deoxygenated blood into this right atrium. Notice there's an indentation here. We're going to come back to that. That's a remnant of a fetal hole that's supposed to be there. And then this is where the circulation from the heart is going to empty, bringing deoxygenated blood that is supplied the heart itself. So that's what those two are. All right, so the blood first enters the right atrium, and then the atria contract and the blood flows to the left, or sorry, the right ventricle. Now notice we're going to talk about the flow as from the perspective of, say, one red blood cell. But remember that while one batch of blood is coming into the atrium and going into the ventricle, another batch of blood is coming into the left atrium and going to the left ventricle. Okay, so the atria are going to fill, and then the ventricles are going to fill. So the right atrium to the right ventricle then contracts and pumps blood out to the pulmonary arteries. Notice this first part right here is called the pulmonary trunk. A trunk is an artery that carries blood usually a short distance and then branches into named arteries. Okay. Now, for those of you who are thinking, hang on, I thought arteries carried oxygenated blood. Is this a vein? No. Most arteries carry oxygenated blood, but an artery is actually classified based on the structure of the walls of the artery and how much connective tissue and muscle. And so these are definitely arteries. Arteries carry blood away from the heart. They're carrying blood that's under pressure. So their walls tend to be thicker. Veins are carrying blood back toward the heart. Their walls tend to be thinner. So I always remember artery away. That helps me remember arteries are carrying blood away from the heart, veins to the heart. Okay, so the pulmonary arteries then, excuse me, are carrying blood. Sorry about that. Are carrying blood, deoxygenated blood to the lungs, the left lung over here, the right lung over here, where the CO2 is going to diffuse out of the blood and oxygen is going to diffuse into the blood. So this is one part of the circulation, and we call this the pulmonary circulation, where this deoxygenated blood goes through the pulmonary trunk to the pulmonary arteries, the right pulmonary artery and the left pulmonary artery, through the pulmonary circulation at the lungs and then back through pulmonary veins, right? Pulmonary veins, notice there are two veins, one artery on either side. And that whole thing is the pulmonary circulation. Then blood returns to the heart through pulmonary veins that empty into the left atrium. So this view that we have up here, this is the back of the heart. So let me go back to here. This is the front of the heart sectioned, okay? And this is the left atrium back here. And you can see these pulmonary veins coming in here. So this view is the back of the heart, and we can only see the little edge of the right atrium down here, right? So we got the pulmonary veins coming in here. From the back, though, you can see the superior vena cava and the inferior vena cava very clearly dumping into the right atrium. Does that make sense? All right, now the left atrium then makes a contraction and pushes the blood to the left ventricle. The left ventricle contracts and pushes the blood out through this valve here. Don't worry, we're going to learn the names of the valves in a minute. Into the aorta. This little part from here to here is called the ascending aorta. And then this is called the aortic arch. All right, now remember, atria do not make powerful contractions. They have thin walls. They're just pushing the blood here to here. That's all they got to do. The ventricle makes really hard contractions, much harder contractions than the right ventricle, because the left ventricle has to push the blood so much farther. So, how does the body ensure that blood flows in only one direction? I'm glad you asked. A bunch of valves. Valves are going to ensure that the blood doesn't flow back to where it's been, that it keeps flowing in one direction. So, both atria fill, they empty, and then both ventricles fill and empty. Okay. Notice that there are valves between the atria and valves between the atria and the ventricles, and then valves between the ventricles and the pulmonary trunk here and the aorta here. All right. The valves between the atria and the ventricles are called the atrioventricular valves. I know, right? Who would have thunk? The right one is also called the tricuspid valve. So it's either the right atrioventricular valve or the right tricuspid, or just the tricuspid valve. That separates the right atrium from the right ventricle. On the left side, we either have the left atrioventricular valve or the bicuspid valve. So tricuspid, this one has 3 parts. Bicuspid, this one has 2 parts. I know they're both shown with 2 parts, but trust me, this one actually has 3. Now, this is a cross-section between the atria and the ventricles. So we can see this is the muscle of the ventricles. We can see the right atrioventricular or AV valve. You can see it has 3 parts, tricuspid. The left atrioventricular or AV valve has 2 parts, bicuspid. And notice the other 2 are called semilunar valves, and they have 3 parts, which is confusing because semi means half. So shouldn't it be trilunar? But no, hang on, I'll explain that one too. All right, the right AV, the bicuspid, the right AV, tricuspid, the left AV, the bicuspid, is also called the mitral valve. This is a mitre, okay? A mitre is also a corner where 2 pieces of, where 2 pieces of wood, for example, come together at an angle. And so where these 2 pieces come together at an angle makes a mitre, and that's where we get the name the mitral valve. Okay, you do need to know all of these names for both valves. I might ask you on a test, what's another name for the tricuspid valve? And you would have to say that's the right atrioventricular valve. Or I could say, what's the mitral valve? And you would need to know it's the left AV or the bicuspid. Okay? All right. Now, the way that you can tell when you're looking at a heart, you can see that the AV valves here and here look really different from the semilunar valves. For one thing, they have these strings attached, and then those strings are attached to these little muscles. The strings are called chordae tendineae, and they're just tendinous cords is what that means. And then these are called papillary muscles. The papillary muscles, papilla, you remember, means nipple. So these are nipple-shaped muscles. I know, lovely, right? There are usually, although not always, 3 papillary muscles in the right ventricle and 2 papillary muscles in the left ventricle. So 1 muscle for each section of the valve. Again, usually not always. The cool thing about these is that when the ventricle relaxes, the wall of the ventricle pulls away from the lumen of the ventricle, and that pulls the papillary muscles away from the valve and pulls the valve open. Then when the ventricle contracts, the papillary muscles move back up, and the force of the blood pushing out together with the movement of the papillary muscles closes the valve. So that's how that valve opens and closes. So the contraction of the atria forces the blood through, pushing the valve open. The contraction of the ventricle pushes on this valve, closing it, and the blood has to go through the semilunar valve. All right, so the pulmonary semilunar valve is between the right ventricle and the pulmonary trunk. The aortic semilunar valve is between the left ventricle and the aorta. And these close as the semilunar valves relax, or as the semilunar valves close as the ventricles relax. Notice the semilunar valves don't have chordae tendineae. The chordae tendineae are only for the AV valves. The semilunar comes from this little cupped shape of these valves, which is shaped like a half moon. Semilunar, luna means moon. So each of these little parts of the valve is shaped like a little half moon when you look at it in cross section. So what happens is, as the blood gets pushed out through the valve, once it pushes open that valve. And then when the ventricles relax, the blood actually starts getting pulled back because then there's lower pressure down here. As the blood flows back, it fills these little cups and pushes the valve closed. So that's how those work, okay? And the force of the blood pulling back into the ventricle actually forces that valve closed. Okay, so this is what that looks like. So here we've got the ventricles contracting and the semilunar valves are open. The blood is pushing against the atrioventricular valves, pushing them closed, pushing against the semilunar valves and pushing them open. Then when the ventricles relax, the blood flows back against the semilunar valve, closing it, and then the atria contract. Just imagine there's blood here, and that blood pushes open that AV valve, and the relaxation of the walls of the ventricles pull on the papillary muscle, pulling that valve open. So when your heart is beating, it's doing this. Sorry, that's just always amusing to me. Okay, the opening and closing of the valves makes the lub-dub sound that you hear as a heartbeat. And yes, lub-dub, lub-dub, lub-dub is actually the technical terminology for the opening and closing of the valves, or the closing of the valves. The lub is the sound of the atrioventricular valves closing. The dub is the sound of the semilunar valves closing. What this shows is that the sounds are not heard best in the exact spot of the valves, because the valves are actually behind the sternum, because remember, your heart is behind your sternum. So if you're a doctor and you're trying to listen for a patient's heartbeat and you listen where those valves actually are, you actually won't hear very much because the sound has to travel through the bone. Instead, if you imagine that the sound goes through the tube and then keeps going. And so these yellow circles here are the areas where the sound is heard best. So if you've ever wondered why your doctor moved the stethoscope around your chest when they were listening to your heart, what they're doing is trying to hear each of the valves separately. Okay, now there are walls between the chambers of the heart, the one between the two atria, cleverly named the interatrial septum, and the one between the two ventricles is called the interventricular septum. I know, who would have thunk it? This one also has a connective tissue layer in between that insulates the electrical charge between the two ventricles. All right, now the whole heart sits inside a really tough sac called the pericardium. It is a fibrous sac, again, very, very tough, very, very strong. It's actually made up of 2 layers of epithelial tissue with a tiny amount of water in between those 2 layers. The water between the 2 layers helps the 2 layers stick together. Think of the 2 sides of a plastic bag. If you get the inside of the bag wet, where the water is, those 2 sides of the bag will stick together, but you can still move those 2 sides past each other. So this gives a little bit of flexibility for movement as the heart pumps. The 2 layers can move past each other. The pericardium holds the heart and restricts its movement and also prevents it from bouncing when you're like jumping for joy or going for a run. And it also prevents the heart from overfilling with blood. You can imagine that if the ventricles overfilled, they could actually overstretch and become damaged. So the presence of the pericardium actually prevents that. So this brown line here, that is the space between the 2 layers of the pericardium. Notice there's also sacs around each of the lungs. We'll come back to that. Those are called the pleural sacs, and we will come back to those when we do the respiratory system. Okay, so there's also, in addition to the 2 layers of epithelium, there is this fibrous pericardium, which is really the tough part. So we have this outer layer of fibrous pericardium, dense connective tissue, irregular connective tissue, very, very strong. It is attached to the structures around the heart, the diaphragm at the bottom, the aorta at the top, the pulmonary trunk, and the vena cava. Then there's the serous pericardium, the parietal layer, the cavity in between filled with a very thin layer of fluid, and then the visceral layer of the serous pericardium, which is actually attached to the heart muscle itself. This inner layer, think of it as like a balloon. It's 1 layer doubled over, okay, with fluid in between those 2 layers. And both layers are connected to the parietal layer is connected to the fibrous layer, and the visceral layer is connected to the heart muscle itself. And then the cavity contains fluid that is secreted by these epithelial cells in those membranes. Okay, what happens if you get an infection of the pericardium? Yes, that's a thing that can happen. The normal immune response is going to bring extra fluid to the area to carry white blood cells. The blood vessels that supply the pericardium are going to bring extra blood flow, extra fluid, and that fluid then unfortunately accumulates in the pericardial cavity between the 2 layers. This is going to be a problem because now the heart can't expand to its normal amount, and so the heart can't pump as much blood out as it normally would. This is going to lead to pain, shortness of breath, fatigue, because literally the blood isn't flowing through your body fast enough because your heart can't stretch as much as it should. Now, the heart itself is supplied by a set of its own arteries and veins. These are the coronary arteries and coronary veins. And on this diagram, the arteries are in red here, the veins are in blue. There are indentations, sulci, around the heart, one for the coronary artery and vein, and one for the right coronary artery, one for the left coronary artery. The coronary veins are going to bring blood back from the heart, and they're going to run parallel to the arteries. They're going to dump into a coronary sinus at the back of the heart, right below the left atrium, and then that coronary sinus is going to empty into the right atrium. All right, cardiac muscle tissue, we already talked about this, so this should be review. Remember that the cells themselves are striated because of the organization of the myofibrils. The cells are connected with intercalated discs that are full of gap junctions and desmosomes that hold the two cells together and enable the cytoplasms of the two cells to be continuous. That means that when one cell receives an action potential, it immediately passes it to the next cell without it having to go through any kind of a valve at all, or any kind of a channel, except like an open channel. It doesn't have to go through a gated channel. All right, so how does the heartbeat travel from one part of the heart to another? All heartbeats start in the sinoatrial node, also known as the SA node. That is a special group of heart cells in the right atrium that are autorhythmic, meaning that they beat by themselves. Now, the heart is autorhythmic because of the presence of these cells. The rest of the cells in the heart are not autorhythmic. They have to wait for a stimulus to beat. But these SA node cells, these are the pacemaker, and they will beat by themselves. The muscle impulse, or the action potential, is generated first in the SA node. It spreads across the atria, across both atria, the right atrium and the left atrium, to the atrioventricular node here. This pathway from one node to the other is called the internodal pathway. Then the beat pauses for just a millisecond. From the AV node, those cells delay the impulse, and then it passes into what we call the atrioventricular bundle, or the bundle of His. Not his, it's named for Dr. His, who discovered it. This is a group of neurons that carry the signal down the interventricular septum that splits in the interventricular septum into the left and right bundle branches that go on either side of the septum. And then those left and right bundled branches, once they get to the apex of the heart, they synapse with special cells called Purkinje fibers, which look a lot like, they're just pretty much multipolar neurons. They're specialized for the heart, but they're multipolar neurons. And those are then going to carry the action potential across and up the ventricle of the heart. So the heart is actually going to contract atria top to the center, and then ventricle apex up to the center, which is useful because the opening of the atria is at the bottom and the opening of the ventricle is at the top. So it's actually contracting the blood and pushing the blood toward those exits from the atria first and then the ventricles second. Okay, so SA node first, across the internodal pathway to the AV node, from the AV node to the atrioventricular bundle, to the left and right bundled branches, which go on either side of the interventricular septum, and then to the Purkinje fibers, which carry the signal up through the ventricles. When we look at an EKG or an ECG, which are exactly the same thing, what we're looking at is a measurement of the electricity created by these signals here. Okay, so first a note about EKG, ECG. It was called an EKG, an electrocardiogram. It was first named in Germany, and Germans use a K for the K sound, always, which led to confusions because Americans spell cardio with a C. So there was an EKG that was an electrocardio with a C gram. So that was confusing. So some people decided to call it an ECG for electrocardiogram with a C. They're exactly the same thing. It's just that Americans can't spell. So that's what this is. And what it's measuring is the events of the cardiac cycle. The P wave shows the contraction of the atria. Then the electrical signal through the atria from the SA node to the AV node, and then there's a pause. During that pause, the atria actually contract. Now, this wave shows the amount of electricity being generated, but not whether the charge is positive or negative. This is actually showing the direction of the charge. The way you do an EKG, the simplest way, is you put 3 sensors on a person, 1 on each wrist and 1 on an ankle. And then you measure which probe or which receptor the signal is going toward. So as this signal passes through the internodal pathway, it's mostly going that way, and that is measured by that bump there. Okay, so this is the electrical signal, this is the contraction of the atria, and then QRS is the electrical signal through the ventricles. This ST plateau is the where the ventricles actually contract. And then the T wave is the cells in the ventricle resetting to prepare for another action potential. We don't see a separate wave for the cells in the atria resetting. It gets lost here in the QRS complex. Okay, so that's atria, electrical wave, muscle contraction, ventricle electrical wave, muscle contraction, reset, rest until the next beat. ECGs are done on this special graph paper and a certain number of lines. I think each of these is 1/10 of a second. So you can figure out how fast a person's heartbeat is by how close together these spikes are. All right, now let's talk also about these terms that we use for blood pressure, systole and diastole. Systole is the word for the contraction of the ventricles. When the semilunar valves open, the blood flows into the arteries, and this is blood flowing into the arteries under very, very high pressure. This systolic measurement, which is the higher of the 2 numbers in blood pressure. For example, if your blood pressure is 120/80, 120 is the systole number. This is a measure of the pressure of the contraction of the ventricle, which is creating pressure against the walls of the arteries. That's what we're actually measuring. Diastole is a measurement of the relaxation of the ventricles. So the AV valves are open, the semilunar valves are closed, the blood is flowing into the ventricles, and the ventricles are relaxed. So if you have a blood pressure measurement of, say, 120/80, 120 is a measurement of how much your heart is contracting and how forcefully it's contracting when it contracts. The 80, the diastole, is the measurement of how much pressure remains in the heart even when the heart is relaxed. So if the systolic number is too high, that means that your heart muscle cells are working too hard to push the blood through your body. If the diastole number is too high, that means that your heart isn't relaxing between heartbeats. both of those things are a problem. So either one of those numbers being too high is a problem. Now, normal blood pressure is actually a range, and I think 140 is danger zone, or getting to danger zone for heart, and 90 or 140 systole, or 150 and 90-ish for diastole. If your blood pressure is above that, then that could be a problem. And it's a problem because it's going to weaken the walls of the arteries, because the blood is pushing out on the arteries too hard with every heartbeat. But it also means that the muscles in your heart are working too hard. Now, what about if you have blood pressure that's very low? That means your heart is working very, very efficiently, which seems like a really good thing. And it usually is. The only time it's a bad thing is if you stand up quickly and get dizzy, because your blood literally doesn't have enough pressure behind it to get to your head sometimes. So people who have low blood pressure tend to pass out more easily than people with normal blood pressure. So, if you have low blood pressure, just be careful out there. All right, now let's talk about the fetal circulation of a fetus inside the uterus of the mother. Where is it getting all of its oxygen from? It's getting it all from the mother. Remember, the fetus is essentially parasitizing the mother. So it can't breathe on its own. It gets its oxygen from the mother. And it actually has a different hemoglobin, which more efficiently binds to oxygen than normal adult hemoglobin. And so the fetus actually preferentially takes the oxygen from the blood. Where does the blood get filtered? In a normal post-birth person, the blood gets filtered in the liver. Well, the fetus is still developing a liver, and so it can't filter its own blood. It needs something else to filter toxins and detoxify things out of its blood. So mom's liver is going to do that job. Nutrients are all going to come from mom, and all of these things, the oxygen, the nutrients, and the waste, need to go either to the fetus or back to mom for removal from the body. And that exchange happens at the placenta. So the fetal circulation needs to transport blood to and from the placenta, which is just a big ball of capillaries, maternal and fetal capillaries, where all of these things, oxygen, nutrients, wastes, everything, are going to diffuse from mom's blood into the fetuses and from fetus blood into mom. But the red blood cells don't cross. Okay, so things that are part of the plasma can diffuse across the placenta. Red blood cells don't diffuse across the placenta. So the fetal circulation, we need to get blood to the placenta, from the placenta, and then return the blood to the fetal circulatory system. But we need to bypass, most of the blood doesn't need to go to the liver or the lungs. We need a little bit of blood to go there to help those tissues do their normal development. But the liver and the lungs aren't fully functional yet, so we don't need the blood to go to the liver. We don't need the blood to go to the lungs to be oxygenated. So the fetal circulation is going to have several differences from the adult circulation in order to bypass the liver and the lungs and to connect to the placenta. All right, so the first thing, here's the fetal circulation here. This is the umbilical cord, and here is the placenta, or the fetus's side of the placenta, okay? So oxygenated blood is going to come through here, and the blood going from the fetus to the placenta is traveling through umbilical arteries, okay? This is deoxygenated blood coming from the fetus to the placenta. It's traveling through an artery because it's traveling away from the heart, and it's deoxygenated because it's already been to some of the fetus's tissues. So the umbilical arteries branch off here from two arteries in the pelvis. These are the internal iliac arteries. So this is the aorta that goes down through the trunk of the body, and then down at the sacrum, it divides into two. These are eventually going to go down to the legs, but these are the internal iliacs that provide blood to the lower, the pelvic cavity and the bottom of the abdominal cavity. All right, so the arteries bring the blood from the fetal circulation to the placenta. The oxygenated blood from the placenta travels through a single umbilical vein, okay? So 2 arteries, 1 vein. This is the opposite of our circulation in the postnatal heart and lungs, where we have 1 pulmonary artery on each side and 2 pulmonary veins on each side, okay? Here we have 2 pulmonary arteries and 1 vein. Both way, no, forget what I was going to say. Okay, the maternal and paternal, or maternal and fetal blood don't mix. The plasma mixes, but the red blood cells do not. Okay. Then this is the umbilical vein coming into the fetal body, and here's the liver. Let me go back to here. Okay, so here's the, imagine the fetal body is here. Okay, so this part is the umbilical cord that's outside the fetus's body. Then it goes into the fetus's body. Here's the liver. Okay, the diaphragm would be here, just under the lungs. Okay, the liver, remember, is the filtering organ that detoxifies any toxins. Okay, but remember, we're going to bypass it primarily. So this umbilical vein comes in and it goes kind of behind the liver. And so this is what happens. Comes in and it branches. One part goes to the liver, and then there's a duct that goes straight to the inferior vena cava. So this blood is going to go through the liver to help the development of the liver tissues. And then this little ductus venosus is going to carry about 1/3 of the blood straight to the vena cava, bypassing the liver completely. Okay, the rest of the blood, this oxygenated blood, is going to go through the liver and then exit through the hepatic veins. Hepatic means liver, which are going to carry the blood to the vena cava, the deoxygenated blood. Okay, so now notice we have oxygenated blood coming in here at the vena cava. But remember that the vena cava is carrying deoxygenated blood. This is the other blood here is blood that has already been to tissues in the lower body. So in the fetal circulation, the blood coming back to the heart is actually sort of half oxygenated. Some of it is oxygenated blood and some of it is deoxygenated blood. So once the blood gets to the fetal heart, remember, it doesn't need to go to the lungs. The lungs are still developing. It's not going to be oxygenated at the lungs. It's already oxygenated. We need this blood to get very quickly to the tissues. So the foramen ovale is a hole in the interatrial septum that allows blood to flow directly from the right atrium to the left atrium, and that bypasses the pulmonary circulation altogether. Because remember, the blood is already oxygenated. Now, some blood does flow through the ventricle, out through the pulmonary trunk, to the pulmonary arteries, back through the pulmonary veins to the left atrium. But some takes this little shortcut, zoop, through there. Okay. If you've ever heard of a baby being born with a hole in their heart, very often it's this foramen ovale that didn't close correctly. Usually, as a baby takes its first breath, that changes the pressure inside the lungs, which in turn changes the pressure inside the blood vessels, which causes this to close. I know that doesn't seem like an immediately obvious chain of events, but that's how it happens. Sometimes this foramen ovale doesn't close correctly. If it doesn't close within the first few days, it will have to be closed surgically, and that can be done. It's kind of a big surgery because it's on the heart, but it's a very possible surgery. So that, if you've ever heard of that, that's the most common malformation, is just a foramen ovale not closing. If, on the other hand, a baby has a hole between the ventricles, that one will definitely need to be closed, and that almost always has to be done surgically. Okay, so we have one more bypass, and that is here, the ductus arteriosus. So remember, some blood is going to continue through this regular circulation, but then here, where the pulmonary trunk splits into the right pulmonary artery and the left pulmonary artery, there's this little duct connecting the pulmonary artery to the aorta. So the blood can bypass and go straight into the systemic circulation going to the rest of the body. Okay, so those are the two bypasses in the heart, the foramen ovale and the ductus arteriosus. Now, after birth, the ductus venosus, which is a duct off of a vein, the ductus arteriosus, a duct off of an artery, get it, and the foramen ovale, which is an oval-shaped hole, get it? The names make sense. How cool is that? All of those normally close. So this is what they turn into. The ductus arteriosus becomes the ligamentum arteriosum that holds the pulmonary artery up against the aorta. And you'll see that on some of our heart models in the classroom. The ductus venosus becomes the ligamentum venosum that helps to hold the liver in place. The foramen ovale becomes a fossa ovalis. So it closes, but it leaves a little indentation, and that's that indentation I told you about at the beginning. So just remember, people get really confused and write foramen, okay? Make sure that in the postnatal heart, it is a fossa, which you remember is an indentation. In the fetal heart, it is a foramen, a hole, okay? The umbilical arteries become ligaments, medial umbilical ligaments, and then the umbilical vein becomes the round ligament of the liver that holds the front of the liver to the abdominal wall. And that is the end of our heart lecture.